It's taking its time. There we go. So today we're going to break it again into approximately 3 chunks. We're going to spend about 20 minutes looking at the lab that we set up on Thursday, which you were supposed to submit by this morning, on the one with the dialysis tube in the beaker with the iodine. Then we're going to spend about 25 minutes talking about osmosis and why osmosis is so critical to life. And then third, we're going to show you a second lab. We're going to do a second lab demo, which takes about 20 minutes to show you the demo and get you set up, and you'll do the data analysis on that lab. All right. So those are 3 things we're going to try to do. Just walk through the last lab, give you some new information about osmosis, and then show you the data for a new lab that you will then do the analysis for. Any questions? Last thing I knew about classes, this could all change, but as of this morning, we're still coming back on the 19th for those that are going to be in person. Those that are going to be remote, you'll be joining us every day using this same link and joining us through Google Meets. And we'll have a camera in the classroom that sort of connects you to what's going on in the classroom. So the big difference is in-person students and virtual students will be in the classroom at the same time doing the same things. At least that's as of this morning. It could all change by the end of the week, but that's the last I knew. Okay, so let me share my screen. Okay, if I go over, wait 1 second. All right, so I go over to classwork. I'm just going to open up the lab from last time. You may want to open it back up yourself. Even if you have to unsubmit it, it's okay. So CSF7. And I'm just opening up the document again. All right. So just to put it back into our memory, because it's been since last Thursday that we looked at this, this picture is a reminder of what we did. This picture is a bit of a reminder of what we did. Let's go blow it up just a little bit. So here is our initial setup. Come on, get out of my way. Here's our initial setup. This is what we set up at the beginning of class on Thursday. We took a dialysis tube to represent the cell membrane, and we filled the dialysis tube with water, starch, and glucose. So those are the 3 things we put into the internal environment: water, starch, and glucose. We then placed that cell model, again, the dialysis tubing being the membrane, the starch, the water, and the sugar being the internal environment. And we placed that cell model into a beaker. And in the beaker, we put water and iodine. So that was our basic setup. The question was, could this membrane control what goes in and out of the cell? And if it does, what materials are able to move through the membrane? So then about 45 minutes later, we took a look and there were some changes. So we still had glucose in the tube, we still had starch in the tube, we still had water in the tube, but we now had iodine in the tube. How did I know the iodine was there? Because the tube turned purple-black, which we had illustrated would happen, or we demonstrated would happen if iodine and starch came in contact. Iodine binds tightly to the starch, and it causes it to make a color change from yellow to purple-black. And in the beaker, we still had water, we still had iodine, because there was still a faint yellow color, but we also now had glucose. And we knew that because the test strip tested positive. So there was evidence that some molecules had passed through the membrane, some molecules had gone out of the cell model, and some molecules had gone into the cell model. So that was sort of hopefully what you saw to move forward. We also made these images on Thursday. The before picture was the beaker had a pretty distinct yellow color, and the tube was cloudy white. The cell model was cloudy white. And 45 minutes later, the beaker was a lighter yellow, and the tube had turned sort of a purple-black. So there were some definite changes in this situation. So then your task was to go back and do the analysis. And so I just want to walk through the analysis to make sure we're seeing it correctly. We're seeing it the same way. Again, please don't be afraid to unmute yourself and speak up if you have a question. You can try the chat. I don't always catch the chat, but I will try to. So let me just go down briefly. So number one says, did glucose diffuse out of the cell? Yes. How do I know? Explain how I know. Because the test strip turned green. Test strip was positive. Normally, if we let it, which we normally do, normally we let it sit for 24 hours because there's usually only enough time in a class to get it set up. Then we come back the next day and get the results. Normally, the first test before we leave it sit is brown, and then the next day it's green, which means there's amount of sugar in the tube decreased, but there's still sugar in the tube. 2, did starch diffuse out of the cell? I'll explain your answer. It did not. How do I know? The beaker's still yellow. If starch had left the tube, it would have reacted with the iodine, and the beaker should have a purple tinge to it. It does not. It's still yellow. That shows me no starch is in the beaker. What is the best explanation for the color change that occurred inside the cell? Inside the cell, it is purple-black. That's because iodine must have gone into the cell, bound to the starch, and changed color. Based on your test results, what substances diffuse through the membrane? Well, the ones that we tested, glucose and iodine. I have evidence for those two. Glucose and iodine moved through the membrane. We'll talk about in a minute. Water also did, but I don't have any evidence to back that up. We did not specifically measure water. Lost where I was. What molecules, what substance did not diffuse through them? This is number 5. Which substance did not diffuse through the membrane? Starch. And my evidence is the beaker is still yellow. If starch had left the tube, that beaker would not be yellow. So I have strong evidence starch was not able to leave the tube, the cell model. 6, explain why some were able to pass through and others were not. Really, you need to go back up to this information here about cell size, the size of the molecule, this background information. And if you look at the background information, iodine is the smallest. It's made up of 2 atoms. Water's very close to being, is also very small. It's only made of 3 atoms, H2O, 3 atoms. So water and iodine are extremely tiny, extremely small, where glucose is about 24 atoms. It's a little bit bigger. It's sort of a small medium molecule. And then starch is hundreds of atoms big. It's a very large molecule. So iodine and water, very small. Glucose, sort of small medium. Glucose goes up in the large category. So remember, we said there was microscopic holes in the bag. And what this illustrates is, most likely the way to explain this is, iodine was small enough to fit through the openings and pass into the cell model. Glucose was small enough to fit through the pores and pass out of the cell model. Water would have also been small enough to pass through the membrane. But starch molecules were not able to pass through because they were physically too big. The starch could not get through that opening. It was too big, which is one of the criteria that happens in nature. It happens in real life. Sometimes things don't get through the membrane because they're literally too big. They can't fit through, along with either they have the wrong charge, they have the wrong polarity, but size is one factor. In this case, we were dealing with a non-living membrane. Size was really the only factor that was going on here. So iodine, glucose passed through because they could fit through the microscopic holes. Starch did not pass through because it was not capable of fitting through the microscopic holes. Number 8, why did the glucose flow out of the cell? Oh, wait, I skipped 7. Let me go back. 7 is going, what other substance may have moved? Water is what we're looking for. You may not have been able to see that on Thursday, but if you'd let it set overnight, you'll see there was less empty space. There was less empty space in the tube. And that empty space was filled up because water moved into the tube. So there usually is some evidence that water moved into the tube because there's less empty space remaining in the tube. Number 8, glucose flow. Why did the glucose go out? Because it was going from high concentration to low concentration. Glucose was in the tube. 100% of the glucose was in the tube. 0% was in the beaker. Almost all the collisions initially were going to happen in the tube. Therefore, glucose, some of it was going to bounce through the pores out into the beaker. More was going to go out into the beaker than would eventually come back in. And thus you would see a movement of glucose out. Same with iodine. 100% of the iodine was in the beaker in this case. And therefore, there was a net movement of iodine from the beaker in high concentration to in the cell model where it was in very low concentration. So both cases, 8 and 9, this is evidence that diffusion does move from high to low concentration. Whoops, why did it do that? Let me go back down. Number 10, you're supposed to change the correct answer to blue. The first 3 would all be less than. Glucose, water, and iodine were all less than the size of the pore. Thus they could move through. Starch would have been the only one that was greater than, thus it could not move through. 11 is just flip-flopping. So 11 is saying, what if we put the iodine and the water in the cell model? So our dialysis tube was a yellow color, and we put it in a beaker that was cloudy white because it had starch and glucose. So what if you flip-flopped it? So the beaker would have been cloudy white, the tube would have been yellow. That means after 24 hours or even after an hour, the tube would still be yellow, but it would be a fainter yellow, and the beaker would have turned purple-black. But the same results would have happened. Glucose still would have moved through the membrane, iodine would have still moved through the membrane, water would have still moved through the membrane, and starch would not have. 12 is a practical example. 12 says we eat lots of starches, like when we eat fruits, veggies, breads, baked goods. But those starches are too big to pass through the small intestine. So we use amylase, an enzyme, to digest starch into glucose. Glucose is much, 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 much smaller. And so glucose can diffuse through the small intestine into the bloodstream so that we can bring it to the cells for energy. So based on the findings of this lab, explain why the digestion of starch to glucose is necessary. So if you don't digest the starch, it's too big. It can't leave the small intestine. It cannot get into the bloodstream. The cells get no energy. The cells die. It's the whole point of the digestive system. The whole point of the digestive system is to take those large macromolecules, those large polymers, and break them into small monomers, because the monomers are what we can absorb. They can pass through the membrane. Proteins are too big to pass through the membrane. Amino acids can pass through the membrane. DNA and RNA is too big to pass through the membrane, but nucleotides can pass through the membranes. Starch complex sugars are too big to pass through the membrane, but when digested, the small sugars can pass through the membrane. So really digestion, the whole point of your digestive system is to break your nutrients down into small enough components so they can pass through the membranes and get to the cells. That's really the whole point of the digestive system. Break down those nutrients till they're small enough to pass through the membrane. Finally, summarize the process of diffusion. Diffusion, you should have said something to the effect of diffusion is the movement of molecules across the membrane. It goes from high to low concentration. Therefore, it is passive and requires no cell energy, and it will result in equilibrium. Those are some of the things we would have been looking for. So I wanted to take the time to run through that so you could at least in your head check off that you understood it correctly or didn't. Any particular questions? Again, feel free to throw it into the chat or to speak up. Once again, I'll encourage you, no questions should only mean because you understand what's going on. So takeaways, This was really a model of how a membrane is selectively permeable. Only some molecules can pass through while others can't. In this case, the selection criteria was size. Starch was too large. Everybody else was small enough. And it was also a model of diffusion. Molecules do move naturally from high concentration to low. Glucose moved from high concentration in the cell model to the beaker. Iodine moved from high concentration to low concentration, moving from the beaker into the tube. So those are the things we were trying to model and illustrate do happen. If you really think you messed it up, you're welcome to unsubmit it and fix it and resubmit it with no penalty. All right. Then I want to go to the next part, the next part of the notes. We'll do this. This will take about 20, 25 minutes. Then we'll take a little break, and then we'll come back and do the lab part of this. So for this part, if you're not already opened, open up the Google. I got to share here a second, sorry. Open up the Google Classroom, and we are going to want to go back to CF6, which you'll see is due before coming to class on Wednesday. So when you go back to CF6, open up the notebook. You should already have slides 2, 3, 4 done. If for some reason you missed that, the lecture is online under Google Classroom. So if you missed it, right here, recordings. This is where all the recordings for each class period are. Right. Links to the Amoeba Sister videos are right here if you need them. Reminder to do the Amoeba Sister videos. I encourage you to do the first 2 before coming to class today. It wasn't required, but I encourage you to. If not, you'll need to go back and do those before Tuesday. And we're going to be picking up here on slide 8. We'll be picking up on slide 8. Note, after slide 8, there's a third video. And there's another reflection. This page right here, page 11, there's nothing you need to do with page 11. I'll be talking about page 11 on Wednesday. So don't worry about page 11. You don't have to do anything to submit it. Just you can submit it without doing anything to page 11. You should do the final reflection, though, on page 12. Right? So all pages should be completed before Tuesday and submitted. Anyway, let's go back to 8. That's where we're going to be spending the next 20 plus minutes, 25 minutes. And then we'll take a break and go from there. All right, I'm going to bring up the actual presentation. That's not what I want. That's what I want. You won't see this slide in your notebook because there was nothing to write on it. So what we're going to be focusing on for the next 25 minutes is osmosis. It is one of the 3 types of diffusion. It's one of the 3 types of diffusion. There's simple diffusion, where the molecule goes directly through the phospholipid bilayer, which again, typically are only non-polar molecules. There's facilitated diffusion, which is much more common, where molecules have to pass through a protein channel. And those are both passive and energy-free as far as the cell is concerned. And the third one we mentioned was active transport, where you do spend energy because you want to go against the natural flow. Osmosis is an example of diffusion, except we're specifically talking about the movement of water, because the movement of water in the body is critical to life, absolutely critical to life. And thus we give it its own name, osmosis. But it works exactly the same. It goes from high to low concentration. It's passive. It requires no cell energy. Typically, it involves a protein called an aquaporin, and it can result in equilibrium. So let's go forward. It says water is very important, so we talk about water separately. Again, osmosis is simply the diffusion of water from high concentration to low concentration across the semi-permeable membrane. So I'm going to take this down just for a second so I can get rid of this box. All right, there we go. Bringing it back up. So down at the bottom of the page, there is what's called a U-tube. Long before there was internet and YouTube. So U-tube because it's literally shaped like a U. So this is typically a plastic or glass tube. And in the center of the tube, my cursor is not cooperating, so I have to describe it. In the middle of the tube, you'll see it's labeled selectively permeable membrane. There's a little membrane disc that separates the left side of the membrane, or excuse me, the left side of the tube from the right side of the tube. You'll see in the first picture, the water levels are equal. You have the equal water levels on the left and equal water level on the right. What's not equal is the molecules, what we call the solute. And here's the key to water. Water movement is affected by what you dissolve in it. Water movement is affected by what you dissolve in it. And you may recall back in middle school, elementary school, you probably did some lessons about solutions and mixtures and colloids. And you probably learned solutions occur when a solute is dissolved in a solvent. Solutes are usually the crystal-like particles that will dissolve in the solvent. In our bodies, the solvent is almost always water. And solutes would be things like salts and sugars and so forth. And so a somewhat, I don't know if you call it practical example, when you were a kid or even maybe now, you made Kool-Aid. I don't know if people use Kool-Aid anymore, but I think a few do. Kool-Aid, the crystals in the package along with the sugar crystals, that's the solute. You pour the powder, the solute, into a container. You then add the solvent, which is the water. You stir it up, and now you have a solution called Kool-Aid, which you then drink. So water is the solvent. The solute is the powder or the crystals, if you would. In our body, our bloodstream, particularly the blood plasma, is 90% water. Blood plasma is 90% water. It's actually the workhorse of the bloodstream. The red blood cells and the white blood cells, they're important, but they do very specific jobs. The plasma is the workhorse. It does the bulk of the transporting. And it's 90% water, which means most everything you put into your blood has to be able to dissolve in water. And what you dissolve in your bloodstream definitely affects your health. So it is inversely proportional. If you increase solute, if you increase the solute, you're going to decrease the water concentration. The more solute you put in, the lower the water concentration becomes. And vice versa, if I decrease the solute, the water concentration goes up. So in the model here, on the left is where I have higher water concentration because there's less solute. There's a higher water concentration on the left because there's less solute. On the right, there is a lower water concentration because there's more solute. What's happening is, let's say this is a water molecule. And what's happening is water is attracted to the solute. If you have a solute in there like a sugar or a salt, there's a charge and the water can become attracted to the solute and the water sticks to the solute. And if the water is sticking to the solute, it can't get through the membrane. If the water is sticking to the solute, it typically will not go through the membrane. So on the left side, most of your water is not sticking to any solutes because there's not very much solute there, so most of the water can move. On the left side, you've got a lot more water sticking to the solute, and therefore it's harder for the water to move. So the left side has a higher water concentration. The right side has a lower water concentration. So as the arrow indicates that in the tube, the water is going to move from the left to the right because it's going to go from high water concentration to low water concentration. Thus, you see in the second picture, the water column on the right goes up and the water column on the left goes down because it's going from high concentration on the left to low concentration on the right. The reason there's lower concentration on the right, because there's more solute. Questions so far? This creates three different scenarios, three different environments, if you would. So we can have this first picture here. So we're looking, if you look across the top of the boxes, those are representing animal cells. And if you look across at the bottom cells, those are representing plant cells. So the first case scenario, the one on the left, this is like a freshwater situation. Fresh water has very few solutes in it. There's not a whole lot of material dissolved in it. We call this hypotonic. Hypotonic. Hypo literally means below, and tonicity has to do with the firmness of the cell due to the amount of water available. So hypo means below. So you've heard, like, you know, that if somebody falls through the ice in a pond, they can get hypothermia. Your body temperature drops well below what it should, and that can be dangerous. You might have heard of a hypodermic needle. Hypodermic needle is a needle that goes below the dermis, the skin. So hypo means below. In this case, hypo means there's low solute in the environment. There's very little solute in the environment. Since there's very little solute in the environment, you have higher water concentration in the environment compared to the cell. So low solute, high water potential, high water concentration. So therefore, water goes from high concentration outside the cell to lower concentration inside the cell. So you can see in the animal cell, water moves in. In this particular case, it causes the cell to lyse or rupture. Animal cells don't like hypotonic situations because in a lot of cases, that's exactly what will happen. The cells will rupture. In the old days, before we were worried about things like AIDS and that, you could prick somebody's finger, put their blood on a slide, throw some distilled water on there, put it under the microscope, and you could literally watch your blood cells swell up and pop like balloons because they had no way to combat the extra water. Plant cells, on the other hand, this is their favorite environment. Plant cells love this environment because they gain water very easy. Remember, if you look in the center of that plant cell, there's that big central vacuole, that little blue bubble-looking thing. That's the central vacuole. The more water it can put into that central vacuole, the more pressure it creates. As that vacuole gets bigger and bigger, it pushes against the cell wall, and that makes a really strong pressure. It's called, as you can see, something called turgid pressure. Turgid pressure is what makes the plant stand up erect. It's what makes the plant very firm and very stiff, so it can stand up and do its normal thing. So hypotonic, low solute, therefore high water concentration. So when you place cells in a hypotonic environment, they tend to gain water. Animal cells don't like that so much because they can't control the flow of water. Plant cells love it because it gives them very good pressure to keep themselves upright. Second environment's the opposite. Say you have salt water. Well, salt water now has a lot more solute because salt is a solute. And this creates what's called a hypertonic situation. Hyper is the opposite. Hyper means above. You have high solute. So you might have heard someone described as hyperactive. Hyperactive is when somebody has a hard time sitting still. They're always, moving around, can't sit still or wiggling, high activity. That's hyperactive. You can hyperextend a joint. I remember in wrestling a couple of times, I hyperextended my elbow, which means I extended it beyond its normal range. It doesn't feel so good. So in this case, hypertonic means there's lots of solute. Therefore, there's a lower water concentration in the environment. So since there's a lower water concentration in the environment, water is lost by the cell to the environment. In animal cells, that causes them to shrivel, shrink up. In plant cells, same thing. The yellow box is the cell wall. That doesn't do anything because it's not living. But the inside contents, the green, which is the cytoplasm and the organelles, that is shrinking up too. We call that plasmolysis. Plasmolysis is when the cytoplasm shrinks up. So neither plants nor animals like hypertonic situations. Causes them to lose water, dehydrate. It's not good for either type of cell. The third scenario you can create is what we call balanced or isotonic. Iso is equal. Think of an isosceles triangle and what that means. So isotonic, you're in balance. You have as much water going in as you have going out. Thus you see double arrows on this picture. So if you have the same amount of water going in and the same amount of water going out, the cell stays stable. The cell remains normal. This is what animals prefer. Humans, animals, we prefer isotonic environments. Plants, they'll tolerate isotonic environments. They don't like it as well as a hypotonic environment because they are a little bit more flaccid. Flaccid means they're a little bit more limp, a little bit more floppy. Whereas if they're in a hypotonic, they're firm and they are fully erect. In this case, the plant might be a little less firm than it should be. But they'll tolerate it if they have to. So let's take a look at those in a little bit more detail. About 5 more minutes here. maybe 6, 7. So let's look at each one briefly again. So this is fresh water. So this is the hypotonic environment. Hypotonic, typically we're talking about a freshwater environment. And that means you have a high concentration of water around a cell and you have low solute. You may have to add a box for the low solute. Just make a text box real quick. I can't remember if I put that in or not. That was a late change. And cells are going to gain water. Cells are going to gain water. So if you put a saltwater fish in a freshwater aquarium, that fish is not going to survive more than an hour or two because its body is not designed to live in a hypotonic environment. What's going to happen is that fish is going to take on a lot of extra water, and that extra water is going to cause the internal organs of that fish to swell up, and that's going to put a lot of pressure on the organs of that fish, and that fish is going to have organ failure and die within a matter of an hour or two. Because of this rapid increase in water pressure, which is going to put pressure on the organs and cause organ failure. So not a good situation to put a saltwater fish into a freshwater aquarium. A lot of places that do canning and jarring and pickling, they have to understand this concept. You know, what liquid you put in the jar with your product is going to affect what it looks like when the customer buys it. So a lot of products like to use hypotonic solutions because that makes the material stay firm, rigid, and maintain its shape. But if it's too hypotonic, by the time they open the can, it may have all exploded and disintegrated. So you got to know the right tonicity to have in the mixture. An example of an organism that has learned to deal with this, there's the paramecium, which we've mentioned before. The paramecium, their problem is they live in freshwater ponds where they would continually gain water. And if they didn't have a solution, they would end up swelling and bursting, just like this yellow cell here. But as you may recall, we talked about parameciums have a way to counteract that. They have something called a contractile vacuole, which is a vacuole that allows them to pump the extra water out and keeps them from swelling up and exploding. Unfortunately, our cells don't have that capability. And therefore, we cannot be continually exposed to a hypotonic environment. Sometimes we'll do it on purpose. So like if you twist your ankle, you will start to release lots of fluids around the ankle to cause it to purposely swell up. You'll purposely create a hypotonic environment, causing that area to swell up, because what you're trying to do is immobilize the joint so that you can't do any further damage to it. Is another example. All right. So for plants, no problem here. They love the hypotonic conditions. Animal cells, oftentimes it leads to kaboom, rupturing and dying if it stays hypotonic too long. This is just a picture of a paramecium. Again, we've shown you one before. In the top picture, again, my pointer's not working. The top picture, there's that, to the left, there's the big circle. That circle is the contractile vacuole that's full of water. And then this picture below, same location, you see now it's like a star there, little starburst. That's after the vacuole's contracted to push the water out. So the vacuole fills up with water, contracts, pushes the water out. Fills up with water, contracts, pushes the water out. And that's how the paramecium combats the extra water and keeps itself from rupturing. Second scenario is hypertonic. I don't know why this one all came at once, but it did. So in this case, we're talking about something like salt water. You have high solute, low water concentration. So since the outside now has low water concentration, this draws water out of the cells and causes cells to shrink. Example are shellfish, things like clams, oysters, mussels, scallops. A lot of them are in salt water. In salt water, that's going to cause them to shrink, going to cause them to lose water, dehydrate. That could be dangerous. So their solution is they have a siphon or a pump where they're continually taking up water and replacing water as it is. And that as they take up the water through the pump, the water is pushing the salt out of the body and keeping the salt from accumulating. All right. In the, couple other examples. A lot of restaurants you go to, they'll give you free peanuts, they'll give you free chips, they'll give you free salsa, they'll give you free pretzels. Why are they giving you chips, pretzels, peanuts? They all have a lot of salt in them. So as you eat those peanuts, you eat those chips, you're putting a lot of salt into your body, which is going to create a hypertonic situation, which is going to draw moisture out of the blood. Water is going to leave the blood to counteract that. And so the blood literally gets a little bit thicker and starts to move a little bit slower. And that triggers in the brain, that triggers the thirst response. That triggers the thirst response. So what the restaurant is hoping is when you trigger that thirst response, you will buy a drink from them. They're hoping, particularly if you're 21, that you'll buy an alcoholic drink because that's where they make the most money. So the restaurants are somewhat being nice when they give you those free chips and salsa and free pretzels and free peanuts, but the prime motive is to keep you thirsty and hopefully have you drink more than you would normally drink, which means you buy more drinks from them. And for restaurants, drinks are one of the ways they make most of their money. Just as another example. We've talked before, if you're stranded on the ocean, don't drink the ocean water because you'll dehydrate faster. It'll be actually more harmful. So he said the alternative is to drink your urine. Why? The urine is isotonic and it's sterile, and it will at least give you a better chance to survive than drinking the ocean water. So hypertonic, the plant, if it only is happening for a short period of time, can survive, but it's not happy. Same with animal cells. Not good long term. Finally, we get to the third scenario again. Isotonic. Isotonic, you have a balance in the concentration water or the solute between the cell and the environment. It's in balance. Since it's in balance, you are in equilibrium. And as the picture shows again, that means things are moving equally in both directions. So for your bloodstream, if you can maintain an isotonic situation, you got no problem. And our bodies are constantly trying to put our body into an isotonic situation. So hypertonic, hypotonic, isotonic, it's not one's better than the other. It's not one's bad and one's good. They're just three different scenarios. We as humans prefer the isotonic scenario. And so our body is always trying to put us back into an isotonic condition. Just like we're always trying to maintain consistent body temp, we're always trying to maintain consistent pH, we're always trying to maintain isotonic conditions. However, as you eat, drink, exercise, do things throughout the day, certain body tissues can become hypertonic, certain body tissues can become hypotonic, and you just have to reset things throughout the day so they become isotonic again. If you are in a situation where you need an IV, IVs are not pure water. If you give me a pure water IV, that's hypotonic. My cells are going to rupture. You're going to kill me. An IV is a mixture of water, salts, minerals, electrolytes that are in balance with what the normal bloodstream should be. So the reason you're getting an IV is you're not in an isotonic situation. In fact, you're far from the isotonic situation. Usually you've had some type of heat stroke or you've got severe dehydration. So this IV is designed to bring your blood back to an isotonic situation. All right, it's just not just random what they put in there. All right, so those are the three scenarios. Those are the three scenarios. So what we would like you to understand is osmosis is the diffusion of water. Doesn't require energy. What you put in the water, though, affects how the water moves. The more solute you put in water, the lower the concentration of water becomes. The less solute, the higher the water concentration. And water will always move from high to low. This creates three scenarios: hyper, where cells lose water; hypo, where cells gain water; and iso, where cells typically remain balanced. Just a reminder that you will do a follow-up. Oops. Don't forget to do the short reflection of new information. There is a specific video about osmosis. Again, don't worry about this particular page right now. And then don't forget to do the final reflection. All right. Questions? Anything? All right. The lab exercise will really, I think, do a nice job helping reinforce what we just said and hopefully help you with that. Again, I will be posting a recording of this if you need to go back and look at something as well. So it's what, 11:22. Let's come back in about 6 minutes at 11:28, and we'll take about 20 minutes to do the, well, it's 11:23 now. Let's come back at 11:30. We'll take about 20 minutes to show you the lab, and then you'll be on your way for the day. All right, so take a short break to use the bathroom, get a drink, do whatever you need to do. 11:30, we'll reconvene. Okay, 11:30. If you're there, come back on camera, please. It take about 20-ish minutes to walk through the lab, and then we'll let you get on with the rest of your day. Okay, so for this last portion, again, this is a lab that normally you would have set up and done yourself. It's a lot more interesting when you're engaged in the setup and the collection of the data on your own. But under the circumstances, we're doing the best we can. So hopefully starting next week, we can start doing some stuff in person again. So for this one, you're going to go to Google Classroom, very top assignment under the week of January 4th. I know technically we're now in the week of January 11th, but first and second hour, we started it under January 4th. So I'm going to keep it there. Anyway, open up CSF 8 and open up the Google Doc. You're going to be filling some stuff in. So I'm going to disconnect the headphones, which will turn off my mic for a second. All right, you should still be able to hear me, yes? Okay. And I'm going to move towards the lab table and share with you some things with the lab setup. So I'm going to show you the setup. The setup actually is pretty simple. Wasn't anything too complicated. I'm going to give you the data that I collected, running the lab for you, unfortunately. And then from there, I'll let you deal with the analysis, which is the most important part. That's where the thinking takes place. All right, so let me first just start with the introduction. If you'll follow along, I'll just quickly read that introduction. It says, using the principles you learned in the first exercise, we will now investigate the movement of water in and out of a model cell. So again, we're going to be using dialysis tubing to model the cell membrane and to create a cell. The passive transport of water across the cell membrane is called osmosis. Osmosis is a form of diffusion. It is just the special case of the diffusion of water across a membrane. In osmosis, water moves from areas of higher concentration of water to a region of lower water concentration. Water is at its highest concentration as distilled water or pure water. So distilled water, pure water is 100% water. There's no solute, zero. The concentration of water decreases as materials are dissolved in the water. And that's the key. The more you dissolve in the water, the lower the water concentration becomes. And you've got it, that makes it hypertonic. If a model cell like our dialysis tubing is filled with salt solution and placed in a beaker of distilled water, think about it, salt is in the cell model, the beaker is pure water, so where's the most water? The higher water concentration is going to be in the beaker because that's 100% water, so water is going to move into the tube where there is some salt. So as it says, water will diffuse into the cell until equilibrium is reached. At that point, no net movement of water will occur between the cell and the beaker. If a model cell is filled with distilled water and placed in a beaker of salt water, now you have the opposite. 100% of the water is in the tube, so water is going to leave the tube into the beaker, causing the tube to shrink. Water will diffuse out of the cell until equilibrium is reached, yada, 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 yada. Then there's the procedure. I encourage you to skim through the procedure later, just to get a feel for what was done to set this up. It's not complicated. I'm going to try to show it to you anyway. I also suggest before you get too far into the data to do the prediction. So for the prediction, again, you're just going to click here, hit edit. So and all you're predicting is which way is the water going to go. Now, key point, hear me. Otherwise, you're going to be confused and get frustrated. The only thing moving in this setup is the water. Water is small enough to pass through the openings in the tube. The other thing we have here is sucrose. Sucrose is bigger than glucose. Sucrose is actually a couple glucose molecules hooked together. Sucrose is too big. Sucrose cannot pass through the membrane. That's important. Sucrose cannot pass through the membrane. Only the water can move. So if the bag loses mass, it's because water left. If the bag gains mass, it's because water came in. The sugar does not have the ability to leave the bag or enter the bag because the molecules are too large. Really key to keep that in mind. So if I look here, my first prediction, I've got a tube that's got 0.9 moles sugar. You'll see the next tube has 0.3 moles sugar. It doesn't matter if you know what a mole is or not. What this just means is 0.9 is 3 times more sugar than 0.3. This has 3 times the sugar of this. This has 3 times the sugar of this, which has no sugar. So keep that in mind. So I make a prediction. I bring this arrow up. Whoops, I got to hit edit. I bring this arrow up, and I'm going to spin it one way or the other. So if I think water will leave the bag with the sugar and go into the cup, which has pure water, I would put the arrow this way. If I think it's going to go the other way, I would spin the arrow to go this way. So you'll go ahead and you'll make your best predictions based on the understanding. All right. So you'll have a prediction for each of the cups. Each scenario is described below the cup. So I encourage you to do that before you get too far into the data analysis. Then day 2 would have been, so I set this up, I actually set this up on Thursday of last week. It has to sit for at least 24 hours. So this is day 2. This is what we do the next day afterwards, which I did do on Friday. You're going to collect the data and you're going to calculate something called the percent change. Okay, so let me show you exactly sort of what we set up. So you might want to pin me for a moment just so you get a bigger screen and you can see what's going on. All right, so what you can see is I've got 5 beakers set up. Those 5 beakers represent the different setups. So if I go back to the document for a second, in this first chart, there's beaker A, B, C, D, E, and it describes what's in the beaker. And that's what I'm showing you here in a second on the screen is beaker A, B, C, D, and E. So if I show it, here's beaker A, whoops, here's beaker A, beaker B, beaker C, beaker D, and beaker E. I have tin foil on them, mainly so that as they sat overnight, water was not evaporating and affecting results. And because some of it contains sugar, I was trying to keep the flies out of it. So that's all the foil did, is it kept it from evaporating and kept the flies from going after the sugar. I'm just going to take those off, and I have my 5 beakers. Okay, again, beaker A, beaker B, beaker C, beaker D, and beaker E. For this, I took dialysis tubing, same dialysis tubing we did the first time. I cut it 5 strips and basically created a very, very similar model. So here's, let me come back up. Here's the one from beaker A. You can see it looks pretty much the same as the ones we created before. I twist, folded, and tied the bottom. I took a syringe, and in this case, added 0.9 molar sucrose solution. So this has got a lot of sugar in it, along with water. And then I tied the top. And then before I placed it in its beaker, which in this case, the beaker is only water, before I put it in its beaker, I weighed it. I got its mass so that I had a way to track whether it was gaining or losing mass. So I took a scale like this one. Well, actually, this is the scale I used. All right. I took a piece of paper towel. I blotted the tube dry so the water on the outside wasn't affecting the weight. And I basically placed this on the scale. And I got the mass, 22.64 here. It's reached equilibrium because the mass hasn't changed. It's only changed a couple tenths since Friday because it's already at equilibrium. Right? So I did that for each of my setups. Here in beaker A, and for each one I've got a sheet of paper. So this was the 0.9 sugar in the tube and water in the beaker. This is the 0.3 sugar in the tube and water in the beaker. And for each one I've got a little slip here that tells me what's what, so I didn't lose track. Whoops, come on. So above the line was what I put in the tube. I put 0.3 moles sugar in the tube. Below the line is what I put in the beaker. That was water. And then there's the starting mass, 18.63. And in this case, the ending mass was 19.831. This particular tube gained about 1.2 grams. I then had a third one, which is only water in the tube, water in the beaker. Nothing but pure water in the model and pure water in the beaker. This one flip-flopped because now I put pure water in the tube and I put 0.3 sugar water in the beaker. This environment has sugar. And this is tube with water and the beaker has 0.9 moles sugar. So that's A, B, C, D, and E. And all I did was measure each tube before putting them into the beaker. And then 24 hours later, I measured each tube again to see if it gained or lost mass. So like I said, the setup's not too complicated. Again, normally you and your lab partners would have been working together. You would have had your own set of beakers. You would have had your own tubes. You would have had your own scale. And you would have done all this on your own, which makes it more interesting than watching me do it. So if we weren't under the current conditions, I would never run a lab like this, where I do it for you, but best solution we've got right now. Questions about anything that was set up? All right, so let me go over here. So here's the setup described, how each one was done, okay? And these are the actual masses that I got. So this is I need you to record these masses on your data table. So I'm going to be quiet here for about 2 minutes. Make it a little bit larger. Be quiet here for a couple minutes so you can type those in. So please make sure you get those masses recorded. You'll need that for everything else you're going to do. And then it'll take me about 5 more minutes to wrap it up. Give me about 30 seconds to 45 seconds. And then we'll get moving. Okay, we okay to move forward? Again, for whatever reason, the recording will be up under recordings in Google Classroom if you need to go back to something. So let me just show you the calculation, and then I think we'll be able to be wrapping up. Wrong one. Let me get the correct one. So there is this data table for class data. We are not using this table, unfortunately. Normally, you would have been in groups of 3. There would have been 9 or 10 groups. Everybody would have had their own data, and we would have taken all of the class data, put it in a spreadsheet, and come up with a class average, which of course would allow us to have a much bigger data set and much more accurate data. But because I'm the only one that could do it, this table will remain blank. So bottom line, this table remains blank. Do not worry about it. Here is the same picture. This is after you've got your results. After you've got your results, you'll go in and put the arrows, and they may or may not match what your predictions were. So let's talk about the calculation. I want to make a comparison. I can look at this and I can see A, there was an increase, 20 to 23. B, there was an increase from 18 to 19. C remained relatively the same. D dropped from 19 to 17, and E dropped from 17 to 14. So I can see that, but I needed to quantify it. Since not all the bags were the same weight to start with, I've got to do a calculation of what's called percent change. I've got to do what's called a percent change. That way it becomes a fair comparison. So to do percent change up here on page 2, this is the formula you will be using. So it says, note it says if the percent change is a positive number, that means there was an increase in mass. And if it was a negative number, then that means there's a loss in mass. So you can get a positive or you can get a negative number. So let me do the first one with you. I'm going to grab a calculator real quick. You might want to do the same. Just running over and grabbing my phone. So right up here, so I'm going to do beaker A with you. Right up here against the formula. So I'm going to take the final mass minus the initial mass. Notice they're labeled final and initial. Initial is what we had when we started. Final is what we had when we were done. So final is 23.55. Subtract initial, which is 20.36. So I'm taking 23.55 and I'm subtracting 20.36. And that's giving me the number of a positive 3.19. Whoops. Positive. That's not the number I want. I want the percent change. So I'm going to take that number, 0.319. I'm going to divide by the initial mass, and then multiply by 100. So if I take 3.19 and divide by the initial mass, 20.36, it equals, again, 3.19 divided by 20.36, hit equal, and then multiply that by 100. That gives me 15.6. I'm going to go nearest hundredth. I'm going to round 15.67%. So it had a positive change of 15.67%. That means that tube after 24 hours got 15.67% bigger. That's what that one means. Take another one down here. Let's look at letter D, and then I'll let you do the other 3. Letter D, same formula. Take the final, 17.57, right here, 17.57. Subtract the initial, 19.4, and I get negative 1.83. Again, I want the percent change. So again, I'm going to take that number and divide it by the initial. and multiply by 100. So negative 1.83 divided by the initial 19.4. So negative 1.83 divided by 19.4 times 100 comes out to negative 9.43%. That tells me this tube got 9.43% smaller. So A got 15.67% bigger. D got 9.43% smaller. All right? It's a better comparison doing the percent versus just doing the difference because they all didn't start out the same size. Hopefully that makes some sense. So I'll let you do the other three calculations. Again, the calculation is all right here. Okay. We are again going to skip the class table. Once you've got the data, like I've got the data for number one, I gained 15.6%. So if it gained 15.6%, that means the water must have went into the bag. And the sugar cannot move. So the only way the bag could have gained mass is the water had to have gone into the bag. And so I would do that. And then these would be based on my data. The earlier ones are based on your best prediction. That's why I say do those first. Okay. Then you have the questions. The questions will help you analyze the data. And I think will help you make sense. In the questions, hear me clearly. In the questions, you should use words like isotonic, hypertonic, and hypotonic. Use the correct language. Isotonic, hypertonic, hypotonic, high concentration, low concentration, high solute, low solute. Those should be parts of your explanation. All right. So to wrap up, you need to have two things done before coming to class on Wednesday. Finish everything in the interactive notebook, including the 3 videos. The notes should be done now. And so complete CF6, that's due before class on Wednesday, and then complete the analysis questions and the calculations for this particular lab. So those are the 2 things you need to have done before coming to class on Wednesday. On Wednesday, we'll wrap things up, give you a review to work on probably during class, probably. And then on Thursday, which is a shortened hour, we only got about an hour. On Thursday, we will take our final test for second semester. All right. So those are the 2 things to do. Any questions, concerns, clarifications? All right. I'm cool if you are. Thank you for your time. We'll see everybody on Wednesday.
Are we coming back or no?
We are done.
Mr. Ebb.
Yep.
I just say I'm sorry I missed like 2 out of the classes last week.
Don't worry about it. I know your mom emailed. I'm so sorry to hear what happened.
Okay. Yeah.
So you just take your time and do what you got to do.
Okay, thank you. I got all the papers printed out. I got like a couple of them done.
All right. Yep. No rush on my end.
Okay, thank you.
Yep. You're welcome.